So bacterial growth is a coordinated process of increase in individual cell mass and size and duplication of the chromosome followed by cell division. Now, when you say a coordinated process, okay, I'd like to put emphasis on the phrase coordinated process. It's actually considered coordinated. It's because the entire cycle of bacterial growth is actually a step-by-step process, okay? So one process happens after the other. Now, this entire coordinated process, once you have achieved the conditions, okay, the conditions are favorable for growth and metabolism, eventually this leads to an increase in individual cell mass and size, okay, cell mass and size, because once the ultimate result of this increase in cell mass and size are actually visible colonies. Okay, this is now the moment that you could actually see the bacteria, no, in the agar plate, in the culture media, and we term this characteristic, no, as colonial characteristics, no, physical appearance of the colonies of that particular bacteria once they undergo increase in cell mass and size during this coordinated process of bacterial growth. Then this um, bacterial growth, no, by growth meaning bacteria undergoing a coordinated process. One of these processes also is duplication of chromosome. And this duplication of your chromosome eventually is followed by cell division. Remember, you have learned that bacteria, being a prokaryote, does not do not undergo mitotic cell division, but bacterial reproduction basically occurs via binary fission. Okay? Now, another term to consider when we talk about bacterial growth is this what we call generation time. Generation time is the time required for one cell to divide into two cells. So we also call this doubling time. Double meaning one cell divides into two progeny, no, or two daughter cells, two new cells with the same chromosomal identity with that of the parent cell, because again, this bacterial growth is a coordinated process resulting to duplication. You just duplicate whatever chromosomal or genetic material that the mother cell had. No, same genetic material is duplicated to the daughter cell, such that one generation time or one doubling time will produce two individual cells with the same genetic material. So I mentioned in the previous slide that bacterial culture, okay, or growth and metabolism is actually a coordinated process because it is comprised of several steps, no, several steps, no, by steps. It has to follow a specific order. These steps, these events, no, occurring in bacterial growth has to follow a specific order. So the first, I wanted to look at the diagram shown here. Notice in the y-axis, okay, y-axis is represented by the number of cells viable. I'd like to put emphasis on the word viable. Viable meaning the number of cells that are alive and are capable of division. Okay, so viable cell concentration, whereas in the x-axis, this is represented by the time it takes to complete the entire bacterial growth cycle. Now, the entire growth cycle, bacterial growth, is comprised of four different phases. First phase is what we call lag phase, followed by log or exponential growth phase, followed by plateau, okay, plateau phase, and then this is followed by death or logarithmic decline phase. Now, stationary phase is also known as plateau phase. Then, followed by death or logarithmic decline, log decline phase. Now, what's really important here, what are pertinent here, is that each steps, no, each phases is actually comprised of several events. Say, for example, in the lag phase, okay, this is actually the time wherein there's actually no cell division occurring, as noted by no increase in the slope of the seen in the diagram, no, versus the y-axis. Lag phase is actually a preparatory phase wherein materials needed for bacterial growth and metabolism are procured, no, by the organism before it could actually undergo its cell division, okay, cell growth, no, duplication or doubling time. And once everything's ready, the bacterial cell now enters this what we call logarithmic or exponential growth phase. No, by the term itself, logarithmic or exponential growth phase. This is actually the time now that bacterial cell growth, no, occurs the fastest, no, such that there is actually high amount, okay, high amount of constant increase in the number of cell concentration versus time, no? Increasing higher amount of viable cell concentration versus time, but the rate is usually constant. The growth rate is constant. Now, what basically happens in logarithmic or exponential growth phase, since in the lag phase, you already have the necessary materials needed for growth, okay? In the log or exponential phase, what happens is that bacteria now utilizes these materials, for example, nutrients, ions, and a lot more of these substances needed for growth. These are utilized by the bacteria in the log or exponential growth phase. This is also the time wherein bacteria synthesizes its organelles, no, prior to cell division. Remember, there's just duplication of everything. Then, once all of these nutrients, the elements, necessary elements needed for growth are depleted, okay, chances are there will be no more, available nutrients, no more nutrients will be available for growth. So, some of these cells now, okay, start to die in the stationary phase. That's why you see a plateau in here. In the figure shown, you see a plateau, no more constant increase in the growth rate because the number of cells that are dividing equals the number of cells that are dying. Dying because the nutrients that are available here in the stationary phase are very limited, and not all organisms get the access to these nutrients needed to continue its growth and metabolism. And so they become depleted of nutrients and some of these elements, and so some cells start to die. No? But the key word here when we talk about stationary or plateau phase of bacterial cell growth, there's actually equal number of cells that grow or divide and the number of cells that die. Okay? However, when the time comes that all of these nutrients and elements needed for growth are fully depleted, you have consumed everything that are needed for growth, chances are there will be no more substances, no, that will be available to continue bacterial growth and division. And so cells now start to die. Okay? When that happens, we enter now the phase we call death or logarithmic decline phase. In this phase, there's actually more cell death compared to the number of cells that divide. Okay? Let's continue. So in lag phase, there's actually no growth rate here. No growth rate, zero growth rate. In exponential or log phase, there's actually constant growth rate. I've mentioned this. And in maximum stationary phase, since there is equal number of cells that divide versus the number of cells that die, there's also no growth rate at all. In the decline phase, since there's more cells that die compared to the number of cells that divide, there is actually a negative growth rate. Okay, negative growth rate, as you see the downhill of the diagram in here, shown here. Now, bacterial metabolism, okay, as I've mentioned, needs actually a lot of elements. Bacteria needs a lot of nutrients and some other elements for it to survive. And another a very good example, no, of an element, the bacteria that needs to survive, although not all bacteria, okay, but most, is this what we call oxygen. No? Oxygen is metabolized by bacteria. And we actually have a discussion before that oxygen are converted. No, once it enters the bacterial cell, it is converted into a superoxide radical, and this superoxide radical is detrimental to the cell, meaning it may damage the cell, bacterial cell, may cause death of the bacterial cell. And there is a need, therefore, to get rid of this superoxide radical. Thus now, bacteria has to metabolize the oxygen it has been exposed to. Now, this metabolism happens in what organelle? In the peroxisomes. Okay, you've learned that before, in the peroxisomes. And that happens in the peroxisomes because peroxisomes contain certain enzymes which are capable, okay, which are capable of metabolizing the oxygen. So the enzymes are as follows. We have superoxide dismutase, peroxidase, and catalase. Okay? So superoxide radical is reduced to hydrogen peroxide and oxygen by superoxide dismutase. And then these two molecules of hydrogen peroxide is reduced into water and oxygen by catalase or peroxidase. Okay? Giving us a harmless byproducts of oxygen and water. Now, take note that according to metabolism of oxygen, according to how a certain bacterial cell metabolizes oxygen, we could further classify bacteria into aerobic and anaerobic. So aerobes, these bacteria requires oxygen for growth. Anaerobes, they do not require oxygen for growth. However, there's actually cases no wherein we have to define terms. No, we need to define terms because we have sub-classifications under aerobic and anaerobic. For example, when you say obligate, how do you define the word obligate? When you are obligated, you have no choice but you have to do it. You have to, you must. Okay, that's the key term there. When you say obligate aerobes, no, let's break down the terms. When we say aerobes, meaning these organisms, okay, actually uses oxygen for ATP or energy production. And when you say obligate meaning there should be oxygen for these aerobic microorganisms to produce ATP. Okay, in other words, when there is absence of oxygen, these obligate aerobes will not survive because their only source of energy or ATP is from the oxygen in the environment it has been exposed to. Thus, the term obligate aerobes. Now, when you say facultative anaerobes, let's break down terms. When you say anaerobes, meaning these organisms most likely do not survive in the presence of oxygen, but the term facultative, okay, changes its meaning. Why? Because if this is obligate anaerobes, meaning obligate, no, you must, there must, okay? An organism is considered obligate anaerobes. That would mean, if the organism is an obligate anaerobe, that would mean that once the organism is exposed to an oxygen, it's very sensitive to the lethal effects of oxygen, and so it's likely to die because it does not contain those enzymes which will convert the harmful superoxide radical and hydrogen peroxide products, okay, into harmless water and oxygen. You get that? So again, obligate anaerobes, they survive in an environment without oxygen, and the presence of oxygen is considered lethal to them. But, but, if we replace obligate with facultative, okay, when you say facultative, this would mean that most likely the way that the organism produces energy is through fermentation. In the absence of oxygen, it could produce energy through fermentation, okay, fermentation of sugars and other substances. But once the organism is exposed to oxygen, okay, it's actually insensitive to the effects of oxygen, okay. But the key word here is that being facultative, it's capable of producing energy even in the absence of oxygen. No? Facultative anaerobes. It does not, oxygen does not really affect them that much. No, unlike obligate anaerobes, that presence of oxygen is actually lethal to them. Let's continue. We say aerotolerant anaerobes. It's still anaerobic, meaning the preferred environment for growth is um, absent of absence of oxygen. Okay, absence of oxygen. That's the preferred uh, environment for growth of all anaerobes. But even in the presence of oxygen, it could still survive. It's tolerant to the aero effects of oxygen. Now, you might ask, Doc, what's the difference between facultative anaerobes and aerotolerant anaerobes? Now, your facultative anaerobes may actually use oxygen in its presence and may continue energy production even its absence. It's facultative. It has two phases, no? Two phases in the sense that it's capable of utilizing oxygen in its presence, but when oxygen is absent, it's capable still of producing energy via fermentation. Unlike obligate, even the presence of oxygen is lethal to them. But when you say aerotolerant, they just tolerate the presence of oxygen. They don't even use oxygen for energy production. Okay, I hope you're getting that. Then we have microaerophiles. Microaerophiles are substances. Now let's break down terms. Philes mean love. They are loving. Aero means oxygen or air. No, oxygen. Micro means low or small amounts. These organisms, therefore, loves small amounts of oxygen. They could survive only in an environment with small oxygen tension. An increased level of oxygen is actually lethal to them. Again, most likely it has something to do with the presence of the three enzymes that I have mentioned in the previous slide. So these are the examples here on the right side. You'll have to really memorize yourselves, no, and categorize as to which is obligate aerobes, facultative anaerobes, obligate anaerobes, aerotolerant anaerobes, and microaerophiles. No? There's a need to memorize all of these. Okay, to continue. So we have this mnemonic here that will help you memorize the entire thing. No? So we have obligate aerobes and obligate anaerobes. So when you say obligate aerobes, again, these organisms really require oxygen for its growth and metabolism, and absence of oxygen would mean that these organisms will die because the oxygen is their only source, no, for energy and ATP production. So we have nosy and nagging pests must breathe lots of oxygen. Cardia, Neisseria, and Pseudomonas, Mycobacteria, Bordetella, Brucella, Bacillus cereus, and Legionella. Obligate anaerobes. They are anaerobes, which are obligate, meaning um, they do not require no oxygen for growth, and presence of oxygen is lethal to them. So anaerobes can't breathe fresh air. Clostridium, Bacteroides, Fusobacterium, and Actinomyces. So when the organism, okay, finds its way to our immune system, the organism actually is virulent, no, and virulent meaning it could cause disease. And these virulence of organism is due to production of this what we call toxin. Okay, so we generally have two types of toxin, exo and endo. It's called exotoxin because exotoxins are generally produced by the cell and released. Third, but when you say endotoxin, endotoxin is actually part of the cell itself. It's not produced by the cell, but it's really a part of the structure of a cell. So again, exotoxin is produced by the cell and then excreted. Endotoxin is really a part of the cell. So exotoxins are polypeptides. They're proteins secreted by certain bacteria which causes symptoms of disease. And exotoxins in general have two subunit structure. We have the A and the B subunit. The A is the active or the toxic subunit, whereas the B is the binding subunit. So the B subunit is the one which attaches to our own cells, the human cells. And once attached successfully to the human cells, the A subunit now, the active or toxic subunit, causes now the disease symptoms. So these are examples of exotoxins, okay? So you'll have to note the different names of the toxins, not much more on the organisms because we'll actually discuss these toxins as we discuss the different microorganisms throughout the semester. Now, what's important to note here are the different types of toxins, its biological effect, and its mechanisms of action. Okay, so take note of these portions of the table for examination purposes. Then we have this what we call, if you've noticed, okay, most of these toxins, exotoxins, may come from gram-positive and gram-negative bacteria. So Streptococcus, Gram-Pus, Clostridium gram-pus, Gram-neg, Pseudomonas, and Salmonella, and among others. But another type of toxins are more or less exclusive to this what we call gram-negative bacteria, endotoxins. They are more or less found in the structure of a gram-negative bacteria because they are located in the outer membrane. And you learned in the previous lectures that the outer membrane is actually a predominant structure of a gram-negative bacteria comprised of a bilaminar phospholipid bilayer. Okay? Now, if an exotoxin has subunit A and subunit B, wherein subunit A is the toxic component, subunit B is the binding subunit, lipopolysaccharide has also a toxic subunit or toxic component we call now lipid A. Okay? So it induces the overproduction of cytokines such as tumor necrosis factor and interleukin-1. Thus, these cytokines, this actually causes fever. Okay? Inducing now activation of the complement coagulation cascade resulting in disseminated intravascular coagulation. Now, once the coagulation cascade is activated in the circulation, this likely promotes clot formation, okay, resulting in intravascular clotting or coagulation. So, to compare exo versus endotoxins, okay, are the following. So, exotoxins are common in both gram-negative and gram-positive bacteria, whereas your endotoxins, being part of the outer cell membrane, are components of most gram-negative bacteria. Because remember, your gram-positive bacteria basically do not have outer cell membrane. So exotoxin is synthesized and secreted from the cell, whereas endotoxin, being part of the outer membrane, is not secreted from the cell. No, it's being part of the outer membrane. Majority of its component is actually comprised of lipids, no, lipid. Whereas your exotoxins are secreted; they are proteins. So located in the plasmid, bacteriophage, bacterial chromosome. What's important to note is that exotoxins are highly toxic, no, compared to your endotoxins, and this may elicit several symptomatology once an individual is exposed to these types of toxins. But very pertinent to endotoxins is shock, no, the fever because of, again, cytokines, tumor necrosis factor, and interleukin-1. Shock and disseminated intravascular coagulopathy due to activation of complement and activation of the coagulation cascade. Now, being a protein, being a polypeptide, it's highly antigenic, meaning if it's highly antigenic, high antigenicity, highly antigenic, this would mean it elicits a really high or rigorous immune response. And when there is very aggressive immune response to a certain antigen, meaning it has high antigenicity, and so if it has high antigenicity, it would follow if that there's vigorous aggressive immune response resulting to increased antibody production. Okay? Endotoxins being lipid, no, being lipids, are poorly antigenic, meaning they do not elicit much antibody production. And so, vaccines, again, you can still recall the main um, the main concept of generating vaccines is especially passive vaccines, no, is actually through production of antibodies which will neutralize the antigens in question. Okay? So since your exotoxins are highly antigenic and high antigenicity would mean high antibody production, chances are you're most likely to produce vaccines against exotoxins. But since your endotoxins are lipids and lipids are with low antigenicity, meaning they do not induce much antibody production and so less likely vaccine to be available. Less likely there's vaccine to be available that will combat against endotoxins. Okay? So another differentiating factor is that your exotoxins are heat labile, whereas your endotoxins are heat stable. Heat labile because these are proteins, and proteins in increased temperature are denatured. No? They are denatured by heat. Then examples of typical diseases includes the following: tetanus, botulism, diphtheria, and cholera. Okay, and then meningococcemia, again caused by gram-negative rats. What organism? Neisseria meningitidis. Okay, I don't know if you've heard of Tdap vaccine. Okay, Tdap vaccine will cover tetanus and diphtheria infection. Okay, Clostridium botulism caused by Clostridium botulinum, tetanus caused by Clostridium tetani. Okay, and then diphtheria, Clostridium diphtheriae, then cholera vibrio cholerae, Corynebacterium diphtheriae. Okay, so once you have isolated, okay, remember the main goal of doing culture is to really isolate the causative agent. And once we have isolated the causative agent, we can actually identify that particular organism which was isolated from the patient, from a source. And once we have isolated and identified the organisms, we can now subject this organism, isolated microorganism, into testing in such a way that we identify them, and we perform also several tests such as antibacterial susceptibility testing to know what antibiotic can be used for treatment of the patient. So again, as a highlight of the entire thing, which I have just mentioned, no, we first we do culture. The main goal, the main purpose of doing culture is to actually isolate the bacteria from a source or from the patient. Now, it's really medtechs and physicians actually have a contrasting roles when it comes to treatment, no, treatment of patients, because in microbiology, no, microbiological aspects, because as physicians, our main goal is to actually kill the microorganism which has caused the disease to the patient. But as medtechs, for the physicians to know which antibiotics to give, okay, not just giving empiric treatment, but antibiotics which are really tailored to the antimicrobial susceptibility testing, there's actually a need to grow the microorganism. And growing and isolation of that microorganism is through culture. So culture is when you get samples from a patient, for example, blood. We call it, if we use blood, we call it blood culture. We can use swab, no? We can use tissues or any other samples which might contain the causative agent. We grow it in the lab. And once we grow it in the lab, no, we isolate the microorganisms. We call it culture. And once the microorganism now is isolated, we need to know what is that microorganism. We need to know the identity of the microorganism so that we can further subject this microorganism to several tests that will help us determine which antibiotic now is capable of killing the microorganism. And that is through now, this is what we call microbial identification. Okay? We identify the genus and the species of whatever culture isolates that we get from the sample of patients. Microbial identification. So we do biochemical testing, for example, fermentation of sugar, growth in an agar medium, selective media, and even PCR. Okay, to do PCR to identify bacteria, but it's kind of expensive. Okay, then we have a serodiagnosis. Remember your bacteria serodiagnosis or serologic testing. This is also known as serologic testing. Remember, microorganisms are considered foreign by our immune system. And by being foreign, they are considered an antigen. And an antigen, being an antigen, okay, they elicit immune response. And one of these immune response, that they elicit is through antibody production. Now, increased levels of a certain antibody directed against a particular antigen is the main principle or the core principle behind serologic testing or serodiagnosis. Okay, so I don't know if you've heard about um, dengue 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 combo test, wherein we can identify if the patient, we just identify the presence of this antibody, and we can tell right now and there if the patient has an active infection, active dengue infection, or past dengue, prior dengue infection. Okay, that's serologic testing. Then we have, once we know now the organism, we have isolated the organism, we have identified the organism, through microbial identification and serologic testing, we have now a diagnosis. What's next is to cure the patient. And these are microorganisms, and usually treatment of bacteria or microorganisms, bacterial microorganism, is usually through the use of antibiotics. So we need to expose now the microorganism to different types of antibiotics to know which antibiotics now is capable of killing the microorganism, the bacteria. No, we call it antibacterial or antimicrobial susceptibility testing. Okay? So again, in culture, we grow or propagate the microorganisms using a media. The media now will provide the necessary requirements for growth. No, it will provide, but until the bacteria has utilized these nutrients in the agar for growth, in the media for growth, what's likely to happen is that it keeps on growing, such that you form now eventually visible colonies. And once the nutrients are depleted, no, mahurot na, are depleted, most likely these organisms also will eventually die in the media. So there is a need, no, to really do subcultures. Subcultures is when you transfer a colonies from one plate to another. Okay, so we can make use of gel or liquid medium. Then we have different types of microbial culture media. Okay, so when you say blood agar, literally we're using agar with blood. We added blood here, but some may use horse blood, sheep's blood, or even human blood. Okay, these are non-selective. Non-selective meaning most bacteria, either gram-positive or gram-negative, may grow on this. Now, chocolate agar, this agar does not really contain chocolate, no? But it contains blood that was heated. No? Thus, the appearance of most likely um, colored chocolate because the blood component was kind of burned, no, during the agar preparation. Then we have glucose salts. It contains glucose. It contains different salts, and this is not selective or differential, meaning any bacteria can grow. Then very important to note is your McConkey agar. So McConkey agar is an agar which is selective to gram-negative rods. By the term selective, meaning it does not allow gram-positive microorganisms to grow in it. It's selective because it contains inhibitors for growth of other microorganisms. For example, it contains bile salts and dyes which inhibits growth of gram-positive microorganisms and gram-negative cocci. So therefore, you see a growth in Imaconki agar. You see colonies in Imaconki agar. You could infer that the organism in question is most likely gram-negative rats. Okay. Then nutrient agar, non-selective, any bacteria can grow in it. Thayer Martin is very specific, no, for Neisseria species. Neisseria only because it contains again inhibitors, particularly antibiotics that inhibits growth of other types of microorganisms, except for Neisseria, of course. It's a media, culture media for Neisseria species. So shown here is a list of different microorganisms and the preferred agar for growth, no? Preferred media for growth. Now, there is actually a need for you to memorize this. Although we will discuss this one by one as we go on with the subject, as we lecture different topics of the subject, but as early as now, you'll have to memorize this. Okay? The bacteria that grows in the agar. That ends our lecture for today. Thank you very much and